for a single locus. And then you can have, in terms of SNPs, single nucleotide polymorphisms. And these polymorphisms, as you see here, in a particular gene, you have A here, at a corresponding position, it could be C here. And this difference in the sequence might contribute to a different phenotype. I have given this phenotype in terms of different colors. And you can see this A to C change, this is single nucleotide polymorphism at this particular site, gives you a different color. Let it be eye color or flower color, whatever you call. So this is a different phenotype. And this is allele number 1. It is not just a sequence alteration, this is also related to a phenotype. So you can have both, in fact, and that is more useful than just a sequence alteration. Just a sequence alteration is of little consequence to us. What we are more interested in is the sequence alteration that leads to a alteration in phenotype, and that would be in a real sense the allele. And these markers are differences which are related to functions would be functional alleles. So you can identify such functional alleles, and they are more relevant from the viewpoint of manipulating crops for improvement of traits. And you can see here, this is one allele and this could be another allele. Here, the A remains unaltered, but this particular site is altered, T to C, and this becomes allele 2, and the color is also changed. So in the same sense, you have many different alterations that takes place, and you have multiple alleles created in the process. And this combination of these three nucleotides, and this is all happening because of these three sites. There are three sites of these nucleotides, they are changing. And this is one combination of these SNP, and that is called haplotype. A combination of SNPs is called haplotype. This is one haplotype, and this is the original gene carrying this particular haplotype, giving you a particular phenotype, and this is the red phenotype. And then this is another combination of these three nucleotides. This is GTA, this is GTC, and this is allele 1. This is another phenotype. And then you have third combination, GCA, and gives you another allele, and so on and so forth. So you have many different combinations of SNPs giving you different haplotypes. And in human, these haplotypes have been met, and in a genome-wide scale for many different genes, and these have been associated with many different human diseases, very complex in nature. But in case of plants, you have advantage. In human, the disadvantage is often, that's why it leads to erroneous results. And the disadvantage is that you cannot phenotype individuals in the same environment, because the data that you collect are from different individuals from different environments. Unlike plants, where you have the advantage of evaluating under controlled conditions in the same environment, and the errors due to environmental impact is very, you can minimize to a great extent. Although you cannot eliminate it, but you can estimate what is the error effect of the environmental effect of the error. So that you can estimate and separate it out from the effect of the treatment, right, due to disease. So far more robust, kind of associations you can establish if you have this kind of haplotype structure constructed, and then for genes, many different genes which are so-called candidates or otherwise, and then associate them with the trait phenotype. In the process, you can identify novel alleles, not just a sequence alteration, but you can have in terms of phenotypic alteration because of sequence alteration. So both you can identify and determine. And this is one such example. In fact, we have done this, and we are doing this in rice and also maize and sorghum, where the sequences are available, and then you can design primers and identify the sequence variation, construct haplotype structure, and then associate them with the trait phenotype. And you can see here, this is the original gene, and this is the color carrying a particular combination of SNPs, that is a particular haplotype. This is another haplotype, this is yet another haplotype, this is yet another haplotype. So they are different haplotypes, and they have different phenotype. That's what the colors indicate. And if you measure, for instance, the trait, suppose I assume that this is measuring yield under stress, like abiotic stress, like drought. What is the yield? And you find that in a set of germplasm lines, suppose you have 200 germplasm lines which you have evaluated under drought stress, and you have said that for each of those lines, suppose there are a set of of those germplasm lines, suppose 25 of them belong to this particular haplotype class. Based on sequencing that you determine that 25 of them belong to this haplotype class. And this is the kind of average yield under drought for those 25 individuals. And similarly, you find there are 30 individuals in this haplotype class, and this is the average yield under drought stress for those 30 individuals. and so on and so forth you determine. And then you find and have a test. You can have a test, significance test done, and then say that this is the kind of haplotype gives you maximum yield under drought stress. In the process, you have a functional allele identified for this particular gene, which gives you a particular phenotype that is higher yield under drought stress. And you can have a set of such haplotypes identified for different genes because they are complex in nature, so obviously they are influenced by a set of genes. So each one of them you can identify a functional set of SNPs which will be actually linked to the genes of interest. So in the process you identify new alleles. This is what actually allele mining is, identify new alleles. And this is one example of a use of the germplasm resource to identify new allele for a trait that is powdery mildew resistance. It's not a complex trait, it's a simple trait, Mendelian trait. And this gene was cloned already in case of wheat. And then the sequences were known. What they did is they made a code collection of 1320 germplasm lines in case of wheat. This is the paper from US, published in Proceedings of National Academy of Science USA in 2009. And they have this code collection, and they have to be phenotyped. Obviously, unless you know the phenotype, there is no relevance of your SNP variation. As I said, you may have SNP variation. You can't call them as actually true-sense allelic variation. Although they are, as I said, in case of microsatellite, we call them as allelic variation. But in true sense, they are not really allelic variation because if you don't consider phenotypic variation. And you can see they identified 211 land races which are either completely resistant or showed partial resistance to this particular disease. And once they identified this, and what they did is they had a marker that is PM3 gene was already known. There was a marker linked to this, and then that's actually it's a gene was clone, so gene-based marker. And they found that this particular gene-specific marker was present in 111 of these land races which are showing complete or partial resistance. So what they did, they amplified the segment, then sequenced, and then they did sequence analysis by alignment, and then found out what is those single nucleotide polymorphisms which are available there. And then what they found is that there are 7 of them which are sequence-wise also different. Sequence-wise are also different, meaning that there are 7 different alternate forms of the gene which are also giving different kinds of resistance which are different from what was originally observed. And how they validated in transgenics. You can see these are the lines, resistant land races. A particular land race, you can see there is no powdery mildew. When this particular allele is knocked down by RNAi, so when you can employ RNAi and silence the particular allele, and when it is silenced, you can see how powdery mildew grows on these leaves of this particular land race. So in the process, they functionally validated this particular allele that this is far more and highly resistant than what was existing. And if you silence this particular allele through RNAi, so you can see powdery mildew becomes the pathogen becomes virulent on this particular line. And this is one of the approaches that this summarizes the approach that you can, if you know the gene, obviously you have to have the knowledge of the gene. So, have a pair of primers, and then you can sequence this in a range of germplasm lines which are very diverse and which have been already phenotyped rigorously. And this is the key. You have been told about phenotyping by Dr. Shesa, and this is the key to success in all these that we are talking about, that if you have rigorously and precisely phenotyped the individuals, you know their phenotype, then you can associate the sequence variation that you see in the PCR product from gene and associate them with the phenotype and then identify new alleles. And they can identify 7 new functionally active alleles of this particular gene. That's what you can do if you have a germplasm resource which is very diverse in nature. If you have germplasm resources and if your genes are not known, right, and rather the SNPs are not known, that okay, this is the SNP or functional SNP, they are not known, but genes are known, and if you were to identify those unknown SNPs which would be functionally relevant, and this particular technology would be very, very useful. And we have standardized this particular technique to use in identification of mutation, and we have nice mutant population. Dr. Robin from your university here, Tamil Nadu Agricultural University, is a partner in this program. Dr. Ravindran is also there associated, and they have also identified many useful mutants, and you can see drought tolerant mutants, for instance, salt tolerant mutants, short root mutants, and blast-resistant mutants, an array of mutants they have identified, Dr. Ravin and Dr. Ravindra, and others are also associated in this project. And and those, you can identify what is that mutation in those genes, and even if you do not know, novel mutations you can identify in the genes. You don't have to really sequence to start with. What you do is you can just isolate. This is the TILing. This is targeting induced local agents in the genome. But this technology was utilized to identify the mutations in the genome in a mutagenized populations. And this is you can extend this to germplasm. That's called eco-TILing. When you extend this, instead of mutagenized population, you can extend this technology to germplasm lines. That's called eco-TILing. But basically, the method remains the same. Slight modification takes place. So in this case, what you do, you have a mutant population like M2 and all that, isolate DNA, equal amount of DNA, you can bulk. For instance, equal amount of DNA from 8 different mutant lines you can bulk, this way and also this way. And the process you can create bulks based on 8 samples, and you can see these bulks, they are denatured, and the PCR amplification will take place. Obviously, if you have a gene and you like to identify mutation in that particular gene, so PCR amplify, the denature and renature, and go for digestion using cell 1 enzyme. This is the enzyme which is utilized. This is the nuclease which cuts DNA wherever it is single-stranded. So when you have a mutation in one line, the other line will be normal, and when you denature and renature, these two strands come together, there will be a mismatch because of mutation taking place. If at all there is a mutation that has taken place. And at that mismatch site where the one nucleotide is mismatching, so there the cell 1 would digest because the DNA is single-stranded. In the process, it will be single-stranded. It will be broken into 2 pieces, and that's what you can identify. So this is one piece that you identify here because you label just one end of the DNA, so you can identify this. And similarly, the other end is labeled with another dye, different dye. This is iron dye 700. This is iron dye 800. So you can have 2 different dyes. So one end is detected by this, the other end is detected by this. That means there are different lanes you can see, and this lane, digestion is taking place, right? And one end is getting detected here, the other end is getting detected in this gel. And in fact, the gel is same. Your detection system is by laser, detection methods. So as a result, you can have 2 different, wavelengths and identify both and superimpose into that, and you find this is the same lane in which this is happening. That means this is one sample which carries this mutation. There are 2 samples which carry this mutation, one in this, the other in this. And you can identify where is the mutation that has taken place in this gene. So this is called TILing, and you can employ this to mine alleles from the germplasm. That's called eco-TILing. And you can mine alleles from the mutagenized population. That's what we are doing. We have identified alleles from DREB1A gene, which is a transcription factor, and which actually influences expression of many downstream genes. And this is a drought-inducible transcription factor. It binds to an element which is drought response element, which is there in the upstream region of many different drought responsive genes. And if you see that DNA binding domain getting altered, means there will be alterations in the expression of many downstream genes, or if that the alteration taking place in the site where it senses the signal, the drought signal, so there will be alteration in the expression of many downstream genes. And we have in the mutagenized population, lines which carry mutation in the functional domain of this DREB1A gene. So you can identify those alternate forms of the gene and associate them with the function. That's what we have phenotyped recently. Some of those lines which are mutated in the DREB1 gene locus at the functional domain, and we find that is alteration in phenotype. And that is how you can identify newer alleles of the genes, and genes like transcription factors which are having influence on a set of genes and a number of genes which are operating downstream in the pathway of drought tolerance. You can employ the sequencing technologies, the next generation sequencing technology, and now the 3rd generation sequencing technologies, like passively biosequencing technology, like ion torrent sequencing technologies, which are newer ones which are coming to market very recently. In fact, passively bio and ion torrent have been launched, and in India probably 1 or 2 machines of ion torrent have been already established, already purchased. So, but these are the ones which have been put to use, to a great extent in many different systems, and to detect SNPs, and then subsequently you can associate them with the trait. The power of this technology is that you don't have to really do gene-wise. Just take one gene, PCR amplify, and identify the sequence. You can do at the genome-wide scale. Suppose you say that you have 200 transcription factors which would be supposed to be involved in drought tolerance. And you can do targeted sequencing of all those 200 transcription factors in a set of germplasm lines, say 500 germplasm lines, and within no time, in a week's time maximum, you can do that. So targeted sequencing of those 200 transcription factor genes, and then see what is the SNP, and then associate them and identify functional alleles for each one of them, and then see which one of those would be relevant actually. So this is the one which is being utilized now to go for large-scale allele mining. In contrast to what I said in case of powdery mildew resistance, that you can PCR amplify one and then sequence that segment by Sanger's method and then identify alleles. You don't have to really do that. You can do that at genome-wide scale and identify novel alleles, and through association analysis you can see how functional relevant they are. You can also employ this particular technology, Illumina breed array technology or Infineum assay technology, to identify novel alleles and a combination of those SNPs and then haplotypes, and identify and associate them. And this is one which is being followed in human to a great extent. In fact, many. If you see Nature Genetics, in every issue of Nature Genetics, you will find some papers published on association analysis using this particular technology. How it works, I'm not really going to details. Going to be 1 hour now. I have to quickly finish it. So, in fact, if you know the SNP site already, and you have to genotype a large set of germplasm lines quickly, then you can do that. And that to thousands of genes, not just 1, 2 genes, thousands of genes. Genome-wide scale association analysis and identify new alleles, and that you can do for all the candidate genes known. All the candidate genes known. If you have 5,000 candidate genes in rice for drought tolerance or drought responsive, you can design these assays for all the 5,000 genes and go at a single go and identify all those SNPs which are showing variation, and construct haplotypes and then associate them with the trait expression. And this is one example. Suppose this is A here in one of the lines, this is G here in another line, and this is supposed to be the heterozygote, if there is a heterozygote available. And you have this kind of this. So this is your probe attached to a bit, and this is the DNA which comes from a particular line here. So you have denatured and allowed them to allele. And this is another line which is contrasting one, having G here, and this could be a heterozygote here. And you have to extend this. This is called single nucleotide extension, single base extension that takes place. And you can, nucleotides can be labeled with different dyes. And you can hear rather biotin in this case. This is nitrophenol. They are different labeled biomolecules. You can see. And this goes and binds here. You can see here there was A here, T is binding, A here, T is binding, and this is biotin, labeled nucleotide, which is extending this particular nucleotide, and then extending this. And similarly, in this case, it was G here, so obviously the C would go and bind here. This is labeled with dideoxy. This will not be extended further. So that's the point. That after this extension it stops because they are dideoxynucleotides. So whatever is extended, it's incorporated, you should be able to detect it. Whatever is incorporated, you should be able to detect it. How do you detect? By different color reactions. We know the biotin reacts with avidin and streptavidin. It's a very strong reaction between biotin and streptavidin. And similarly, you have an antibody which is generated against this particular one. And you can create this kind of one, particularly say for instance this one anti-DNP red, and this is streptavidin green. So they are differentially colored. And this would go and bind here, and this would go and bind here, and this would be binding to both. And that is how green color will tell you what was there, whether it was T there or G there. Because it is green color, it has to be T there, right? Or A there in the original one. So. And since it is red color here, it has to be C there. That means there was G here in the original one. So here there will be a mix of green and red. So obviously it will be yellow. So that's the reason why it has to be heterozygote. So you identify what was the SNP. It was G there or T there, or it was a heterozygote. A particular individual is genotyped in the process. And you can do that for all the SNPs which are there in a gene, or in the genome-wide scale you can analyze that. And once you do that, you can construct haplotype, as I said in the previous case. and then you can associate that with the phenotype and identify new alleles for the traits of interest. And you can image it, for instance, as I said.